The overall goal of this procedure is to provide a standardized approach for sampling environmental and drinking waters for occurrence of enteric viruses based on EPA Method 1615. This is accomplished by first assembling a sampling apparatus, sterilizing the intake and cartridge housing modules of the apparatus, and then aseptically adding a sterile electropositive cartridge filter. Next, a defined volume of water is passed through an electropositive filter contained in the sampling apparatus. Then, the excess water is drained from the apparatus and cartridge filter housing, and the volume sampled is recorded on a sample data sheet. Finally, the sampling apparatus components are packed in a cooler shipping container kept at 1 to 10 degrees Celsius and delivered to an analytical laboratory. Ultimately, enteroviruses and noroviruses in environmental and drinking waters are measured using this sampling procedure as part of an overall method.
This procedure uses an electropositive filter to concentrate viruses in environmental and drinking waters. There are several other filter types that can be used to do this procedure. One is the ultrafilter and another is a negatively charged filter. The advantage of the electropositive filter over the ultrafilter is that the sampling is about 2.5 times faster.
Virus collection using negatively charged filters requires pH adjustment and the addition of salts. The advantage to positively charged filters is that no adjustments are needed prior to sample collection.
Generally, individuals new to this procedure will struggle with maintaining sterility and with unexpected conditions, such as the absence of a convenient tap for sample collection.
Demonstrating the procedure today will be myself, Shay, Fout, Jennifer Cashdollar.